I think Samir Parikh's group actually presented some very interesting single cell, sequencing data in post-BCMA therapy patients. I thought that was quite interesting. Again, hypothesis generating, but do you have any insights you can share about that data?
So they, yes, Samir Parikh's lab did presented several very interesting abstracts focusing on post-BCMA therapy when people relapse, whether it be post-BCMA with a bispecific or post-BCMA with CAR-T, what to do in those cases. And they collected all the data of patients that have been treated at Mount Sinai and received different either bispecific or CAR-T after having a BCMA relapse, and saw that there is response and there is feasibility of sequencing bispecifics in people after they relapse from one bispecific or from CAR-T therapy, even if it is from the same target, whether it be BCMA or if it's a different target like GPRC5D or FCRH5. And now that there's new targets being evaluated, it's just kind of increased the ability to be able to transition from one to the other, or maybe to even combine to see if that can actually improve resistance and progression. The other interesting thing that it showed is that one of the concerns that we have with bispecifics and CAR-T and the relapses, could it be because of T-cell exhaustion? But if they are responding to a different bispecific agent or a T-cell redirecting therapy, then that tells us that it's not that there's T-cell exhaustion. It might be something related to the antigen we're actually targeting in the myeloma, that there might be shedding or underexpression, and switching to a different could be an option, and not necessarily concerned about the T-cell exhaustion. So that was a very interesting revelation from the studies that were presented at ASH this year, that I think really opens our eyes as to how we can now either sequence or combine the T-cell directed therapies so that we can have optimal response from them.